just to introduce Ross Jones very quickly, that we're very proud to have here. So thanks, Ross, for coming, and thanks a lot for stepping in so short time, of course, also like you could have come to do some days ago. So just a very brief recap for Ross CV. So he got his bachelor in bioengineering at the University of Washington. Then he made his PhDs at MIT in bioengineering, if I'm correct. And so he's currently a postdoc and was awarded a postdoctoral fellowship from the Michael Smith Foundation for Health Research. And I'm very curious to hear about his work on engineering mammalian cells. Thanks a lot.
Great. Thank you, everyone at this conference, for the patience. Can you guys see the slide okay and hear me all right? Great. Yeah, a little bit of a mix-up at the time when Domitilla, who was originally going to present this, asked me to present today. I didn't realize she was saying 9 a.m. Eastern time, because I'm here on the West Coast in British Columbia. So anyway, today I'm going to talk about some work that I did in my thesis when I was working with Domitilla and Ron, my 2 thesis advisors at MIT. And this presentation will also prominently show a lot of work that my colleague Yili worked on when he was in Domitilla's lab. And Yili's now a postdoc in the Venturelli lab at University of Wisconsin, and definitely a great future academic prospect for anyone interested. Anyway, thanks again to the organizers, and we'll go ahead and get into this. The slide will advance. There we go. Great. So as somebody on more of the biology side of the synthetic biology and modeling communities that are fusing together for this field, I get really excited about the different applications of synthetic biology for medicine and for biotechnology. So in the short term, there's been a lot of great work from Jim Collins' lab and many others, Chris Voigt, just those guys at MIT, but many beyond at other labs in Boston and Stanford and in Europe, producing different types of technologies with biotechnology already. So whether that's diagnostics for viruses that work with cell-free reagents, using principles of genetic engineering but extracted from cells, or building technologies that can either detect biothreats or make it so that we can use cells more safely within our own bodies, or manufacturing various high-value chemicals for use in industry. In the medium term, we're starting to really be able to, or we'll hopefully be able to gain enough control over cells and communities of cells to be able to do really sophisticated engineering of the microbiome or other communities of cells in, let's say, growing plants or growing other types of organisms, as well as using the ability of cells to sort of home and target different regions of the body to do things like targeted drug delivery to treat diseases like cancer. But eventually what we want to be able to get to is to sort of wholly be able to engineer and control what cells and tissues will do, so that, for example, we can develop tissues that will be able to self-repair and will have many sort of unique and controlled properties that a typical tissue or a typical material wouldn't have on their own. And if we could do this in a sort of patient-specific manner, we could really harness the power of biology to develop cures for diseases, prevent disorders from arising in certain patients that might have genetic abnormalities, or even in the future maybe rejuvenate the health of the people that have had their health deteriorated over time. But to get there, it's going to require a lot of work. One of the challenges is sort of creating biotechnologies that work. And so we'll get into a little bit about why things don't usually work. In computer and electronics designs and sort of the other fields of engineering that have really progressed over the last century, there's been this ability to abstract and compose large systems from small systems. And the benefit of doing this sort of decomposition of a system is that we can sort of build really well working subsystems like transistors. We can compose those into logic circuits. We can put those onto circuit boards or other sort of modular compositions of many different components. And eventually we can build up sophisticated things like this computer that I'm working on. And at each layer of design, we can kind of forget about what's going on at the lower layers because everything is so well designed and so well insulated that there's not going to be too much sort of crosstalk between any of these physical parts and modules within the system. And of course, no computer works perfectly, but we can make really great things with this strategy. In biology, it's really tempting to say, hey, look, we can break down the tissue into the cells, and within a cell, you can look at its pathways and the reactions within those pathways and say, what if we could do the same thing? Just compose this up from the bottom, design the proteins and the genes, and sort of let them emergently give us the behavior that we want. And given our ability to sort of engineer DNA more and more rapidly over time, this should become easier and easier. But the difference here is that in biology, modularity doesn't quite work out in the same way. So instead of modifying one sort of gene regulatory network in the cell and having it stay on its own, it's influenced by everything else going on in the cell, mostly because almost everything in the cell can sort of react with everything else in the cell. So we don't have nice wires that can separate one channel of information flow from another. And this means that when you try to build any sort of sophisticated system, it can take an entire PhD thesis to do it, which I definitely have seen those and have worked on those myself. Okay, so why is modularity such a challenge in biology? Well, one thing is that if you have a couple gene regulatory networks, which we can sort of abstract here as these boxed modules, and we connect them to each other, maybe not even something so sophisticated, maybe just two genes where one is regulating the other. When you connect the one gene to a downstream gene, that downstream gene will apply a load to the upstream gene, meaning that it's the fact that it's using that gene's product, so let's say a protein for regulation, means that it is going to use that gene's product and then reduce its availability to other genes if it's going to try to regulate those. Another key factor that will be highlighted much of the rest of the talk are these cellular resources, so transcription factors and other molecules within the cell that are needed for genes to produce proteins and RNAs and other things. So every gene within the cell has the ability to interface with at least some of these, and that causes their availability to decrease with respect to other genes. And finally, the genes themselves that we engineer, the proteins that we engineer, the pathways we engineer in the cell, can have off-target interactions, meaning that they can regulate things that they're not supposed to regulate through direct action. They could also indirectly regulate things going on by affecting, for example, the growth rate of the cell. And all of this has the effect of sort of perturbing the overall state of the cell and the regulation that should be going on. So it's a tricky system to be working with. So all of these we think of as simply lack of robustness or perturbations that affect any desired engineered behavior that we're trying to establish. And so this is a control design problem. Over the last several years, Dr. Tillow's lab, among several others, has been really working up this idea of fusing together synthetic biology and control theory so that we can design properly controlled systems that regulate their gene expression. Okay, so I mentioned some of the things that are involved in, or that affect the content or the expression of genes, but we can do this a little bit more systematically. So when you introduce a gene into the cell, where you put it into the cell, how it's oriented in the genome, if it's in the genome, or how it's oriented in its own sort of genome, if it's in, let's say, a plasmid or a virus, matters for how the gene will be expressed. And the chromatin state for eukaryotic systems is another layer of regulation of gene expression that we have to keep track of and can affect whether a gene is expressed. And this is a big problem for engineering mammalian systems in particular, because the chromatin tends to get silenced if you leave that gene in there for a long time. It can be a real frustration. We also have to think about the cellular context. So what kind of cell are we engineering? How will the genes that we introduce interact with other genes through some of the mechanisms that I mentioned on the previous slide? And finally, the extracellular context is, of course, important too. What other cells are nearby? How are they signaling and interacting with each other? Are they competing with each other for shared nutrients or space even? And what are the other sort of extracellular matrix and other factors that affect cell growth and behavior? So today we'll just focus on one of these problems here, which is resource sharing or resource competition among genes in the cell. But of course, there's a lot of interesting things to dive into here. Okay, so Domitola's lab's interest in this sort of started with a couple of papers by Andres and Shinho, who Andres was a former grad student in the lab, and now he heads up the lab in Abu Dhabi, and Shinho is a current postdoc still in the lab, also fantastic postdoc if anybody is looking for somebody to recruit to their academic program. So what they started with was looking at resources and how they're shared and affect gene expression in bacterial cells. So if you have several genes or gene modules that are in the cell, they should presumably be using some set of the cell's resources for their growth or for their expression. And so if you have some genes and they perturb the availability of all cellular resources, then you would expect that for some other module, but say one that you weren't engineering at the time, to be affected by this lack of cellular, or this disruption to the cellular resources. And so indeed, what Andres found is that if you induce the expression of some other gene, which isn't being shown in this graph, but is being activated in its expression, and you just look at some constitutive other gene that's in the bacterial cell, that the more that you induce with a stronger promoter, basically you will further and further perturb and decrease the expression of this other sort of standby gene, which is not ideal. Further, and this one was really weird. So Shinho was working on putting together a couple of different genes where maybe this gene should sort of be induced, so turn on in response to its input, and then that gene should further induce or turn on a downstream gene once it is activated. But what he found is that instead of the expected sort of on, off, or turning on in response to the inducer in this case, it was actually turning off, which is really strange. And so it turns out that that was an effective resource competition. And he went through some nice analysis to show that in different sort of parameter regimes of the relative expression of these two parts, that you could fix this just by accounting for the resource competition effect. But yeah, a lot of the state of things in the field at the time was sort of not really thinking about these issues. And so when you would see something like that, you would be quite confused. So from these works, lab came up with a couple basic rules that work for bacteria. So the main thing in bacteria that's limiting are the ribosomes, which are basically being synthesized as fast as the cell can make them. And so it's relatively easy to saturate their availability when it comes to gene expression as well as growth of the cell. So there's a couple of simple design rules you can follow to kind of account for resource competition effects in bacteria in your head. One is that if you have a gene that is activating another gene, that it will tend to have a repressive effect on other genes. That's just simply because of the increased resource usage of this gene, drawing them away from some other downstream gene. On the other hand, if you have a gene that is repressing the expression of a second gene, then that's going to tend to increase the expression of other genes due to the freeing up of resources. So this was some really nice analysis that we did, and has really helped us to understand what's going on in bacterial systems. But we were interested in what are the consequences of resource competition in mammalian cells as well. And so that's where I come in, and I'll talk a bit about my work sort of characterizing resource sharing effects in mammalian cells, and then also building some different control systems to help solve the problem of resource sharing. Cool. So in mammalian cells, the story is slightly different. In bacteria, I mentioned that ribosomes tend to be the most limiting resource for gene expression, but in mammalian cells, it seems like there's a difference, and that different entity that is shared are transcription factors and their co-activators, which are very commonly used for inducing gene expression. So how do we know that? Well, what we had started doing were some experiments like this, where we would have a transcription factor, so GAL4, activating the expression of some target gene. And in another module, we would just have a constitutive reporter. And this reporter would basically tell us if there was any depletion of the shared resources due to the action of GAL4. And so what we see is that if you transfect cells, these are HEC293 cells, a kind of well-used cell line within mammalian cell synthetic biology space, and commonly used for bioproduction, that if you transfect cells with GAL4 fused to one of a couple of different activation domains, which are domains that recruit transcription co-activators to drive transcription, that you can severely deplete the amount of RNA expressed from this gene and protein expressed, measured by qPCR and flow cytometry. And so the qPCR data shows us here that the transcriptional resources themselves are certainly going down, and it's not just an effect at the protein level like what we would see in bacteria. Further, if we went through a series of experiments to sort of look at this in more detail. So looking at the expression of our reporter and our activation reporter, and looked at a few different cases. So one is looking at whether this gene actually even needed to be here for this effect to be observed. So if you don't have this gene here at all, you still get repression of our off-target module when you have expression of the full fusion protein, GAL4 Viper, which is a strong activator, or even just the VPR activation domain itself. And so in this case, there's not even any production of this gene going on. So it's not necessarily the effect of just producing this gene that is sequestering resources or otherwise affecting module 1. It's just the basically the presence of these proteins being made, because we can see basically a similar effect whether or not that gene is there. And just to confirm, we can see activation of this only when we express a full activator. So what is going on? It's a little bit weird compared to the bacterial case, but the difference is, I'll go through it in a little bit more detail in the next slide, but the difference is that here, the activation domain, whether it's on the transcription factor or not, is solely able to bind and sequester transcriptional resources itself, regardless of whether it's actually being used for activation of some downstream target gene. Okay, so what this leads us to is a sort of slightly more sophisticated model of transcription than is typically used. So typically you would just account for the binding of, let's say, a transcriptional activator, and then if it's bound or not, that determines whether RNA polymerase can bind or not and give you transcription. But in eukaryotic systems, because of the necessity of co-activator proteins, we can sort of revise our model to say, okay, a transcriptional activator needs to bind to a promoter, recruit these co-activator proteins, and then drive transcription. But it's also possible for these transcriptional activators to bind to these co-activators in solution. And this seems to be the main problem. So we can model this mathematically by basically accounting for the binding of the co-activators in the model of the expression of this gene of interest here. And the free amount of co-activators we can model basically as some total initial amount that is reduced by the addition of all other transcription factors or co-activators, transcription factors or their transactivation domains that will sequester the particular set of co-activators needed for a gene of interest. So this use here would sort of be any transcription factor that would bind to this resource, but notably it can also be the transcription factor that is used to drive this transcription as well. So that set is not exclusive of any transcription factor that is also an input to the system directly. And so the consequence of this is that if you have a system where you're trying to activate some gene X2 with another activator X1, we would expect for sure that some off-target gene X3 would be repressed due to the decreased availability of these shared resources. But for the activated gene itself, it's actually unclear for a given level of transactivator a priori whether it would be going up or going down in terms of the induction of X2, because of the ability of that transcription factor to also sequester its own resources that are needed for production of its target genes. And so what this looks like in data is the following. So here's a relatively weak activator. When we transfect it into the cells at increasing dosages, it has a very mild effect on the off-target gene here, and it has a nice dose response in terms of activating its target gene, where it just kind of goes up. But if we add increasingly strong activators, you can see that we repress further this off-target module, and then we also have this biphasic effect where if we add more and more of the transcription factor, that eventually the expression of its target gene will go down. So this is not like a wholly new known effect. There's actually some great research from the 90s that was initially sort of investigating how transcription works and worked out the necessity of certain co-activator proteins, and we're seeing some of these effects. But we've just built upon that here to sort of really quantitatively dig into the model of how this works. Okay. So this isn't just specific to one cell line. So just to convince you this is a pervasive problem, we looked at various different promoters here that could be affected by different Gal4 activators in different cell lines. And so we quantified the expression of all these different promoters in different cell lines in the absence of any module 2 here, or the absence of any activators being added. And then we computed the full change of all of these levels once we added in these different activators in different cell lines. And so what you see in general is a lot of purple, which means that the genes are being repressed. There are a few cases of green, which was weird to us for a very long time because how were the genes going up? Turns out that most of these are instances where actually the transcription factor can bind to these promoters with some weak affinity, because this is not the most precise transcription factor, and it can bind to GC-heavy promoters like these ones. And there's also an effect, going back to what I said about growth rate feedback, where some of these activators are kind of potent and affect the growth rate of the cell, and for the promoters that they affect less strongly, like UBC, if the growth rate of the cell decreases, then the overall effect of that, or the overall effect on that gene, will be for its expression to go up. So we eventually kind of worked out how some of these things could be going up. But I think that just points overall to the difficulty in sort of predicting what the effect of one gene on another will be. We checked other transcription factors as well, and were able to see we didn't check them in all of these different cell lines, but besides Cal4, we looked at other transcription factors and saw that they were also able to repress other genes. So we need basically a way to more robustly control gene expression so that in different cells and in different contexts, the gene expression can be maintained and will not be perturbed by everything else. So in that vein, I'll talk about a couple of control systems that we worked on to try to do just that. So for any given genetic device that we put into the cell, which is just one gene that produces one usually protein output, resource competition we can just think of as some disturbance input to the system that affects the gene expression, so that when there is this perturbation that occurs, the expression goes down. And if we could just abstract it like that, then we can think of control strategies that are able to take the system and make it robust to these perturbations. So a really common engineering strategy to do this is negative feedback control, where you detect the level of output being produced by some device of interest, feed that into a controller, and then use the detected difference between the output and the desired input, which is given into the controller, to update the expression of the system. Another way that we can do it is through feed-forward control, which is a way of basically directly detecting the disturbance. So something that can respond to resource competition, for example, in a similar way to the device of interest, and then using that to basically offset any perturbations that occur to the device itself. The first, whoops, got ahead of myself. So if these systems work well, then instead of having some sort of depletion of gene expression in response to a perturbation to the resource availability, the system would return to its desired output state. So first I'll talk about the negative feedback design that we did, and then I'll get to the feed-forward one. So Yili and Domitilla several years ago worked up a strategy to deal with resource competition and other effects through negative feedback that implements what we call quasi-integral control. And so for those that are familiar with integral control, I'll explain what this quasi-integral concept means, because it's specific to systems like cells where it's not such a perfect environment for doing feedback control. Okay, so let's say we had our device and we wanted to plug in an integral feedback control into it so that we can control expression levels in the face of perturbations. How would we do it? Ideally, with an integral controller, what you're doing is that you're measuring the difference between the input and output, and then you're integrating that over time. And so the strength of the input provided to the genetic device is then proportional to the integral of the difference between the input and output over time. And if that difference continues to exist, the integral control becomes stronger and stronger until it returns the device exactly to its set point. Because the steady state of this controller is only zero, or it only reaches steady state rather, when the output of the device will exactly match the set point of the controller. However, this doesn't work perfectly in human or in any type of cell, because the cells are growing and dividing. And also beyond that, the molecules that we use to implement these systems are decaying due to spontaneous degradation and other mechanisms. So we have to account for that this controller isn't just some machine in the cell. It is itself implemented with molecules. And if we just take it as, let's say, one gene or one protein molecule, that protein molecule has some decay rate, making it so that it's not just going to be representative of the difference between the input and the output, but it will also have this leaky term here. However, if you can increase the speed of the controller, so make basically the reactions implemented by the controller very fast, to where this has a very high gain, then what you can get is a situation where the controller can return to more ideal integral-like behavior, where basically the reactions of the control system have overwhelmed the decay kinetics of the control molecule itself. And so in that case, we can somewhat ignore these decay kinetics. So we need a system, basically, that can implement very fast reactions within the cell to be able to do this. And what they came upon as a good way of doing this, and in Yuli's paper, he looked at a couple different examples of systems. So the one that we decided to implement was this one, so I'll talk about it here, was phosphorylation reactions. So phosphorylation is a mechanism, for those that aren't familiar, in cells where proteins can be modified with a phospho or a phosphate group. And the effect of this is usually to modulate the functional activity of the protein. And so if you have, let's say, a transcription factor that is phosphorylated, which we denote here with this asterisk or with a P in future slides, it might be annotated that way, that allows it to be in an activated state where it can then induce gene expression of some downstream gene. If we make it so that that downstream gene produces some kind of regulator that can dephosphorylate this transcription factor, then we'll have created a negative feedback loop where this entity here, the phosphorylation cycle, is considered the control system itself. The input is the kinase that converts the transcription factor to its phosphorylated form. And then the output is some reporter that we can detect, but also we would produce some phosphatase that dephosphorylates this protein. So if you write out the reactions for these using sort of standard Michaelis-Menten models for the enzymes that are doing these phosphorylation and dephosphorylation reactions, we get a system that looks like this. So X star is the key operative transcription factor here. And this is the expression of the output dependent on the level of X star and not X, which is the not phosphorylated form. So under the condition where these two phosphorylation and dephosphorylation reactions are in a saturated state, so the enzyme is working, the enzyme levels are very high. And if the kinetic rate of the enzymes is very high, so we have a very small epsilon value, then we can get this sort of model where the expression level of the transcription factor here, the phosphorylated form of it, will only go to steady state when, if this epsilon is very small, when the U and so the input and the output are balanced. And we'll just note that instead of Y equaling U exactly, there is this proportionality factor in front of U that accounts for the kinetic rates of the enzymes. So this will be stable as long as basically this reaction here has not been saturated. So the transcription factor can't have saturated its target promoter, otherwise a change in the amount of the phosphorylated transcription factor won't have an effect on the expression of the reporter or the phosphatase output critically. So what we expect is that for some open loop version of the system where you don't complete the negative feedback loop, if you add perturbation to resources, that we will decrease expression of the output across the sort of input-output space of the system. But if the control is working correctly and we have the system set up in the closed loop format, that across the input to output space here, we should be able to maintain that when we add perturbations. So to implement this, we turned back to the bacteria. So mammalian systems, eukaryotic systems in general, have many different signaling pathways in the cell with a lot of different types of kinases and phosphatases, phosphorylating and different proteins. And to be able to engineer within that space is very difficult because you're going to open yourself up to a lot of potential off-target phosphorylation effects, which can confound the function of any particular gene regulatory network that you're making. So instead, we look to port a system from bacteria that doesn't exist in human cells and actually doesn't really exist in any animal systems that we know of. So what we ported over are these two component signaling systems from bacteria. In these systems, typically there's some kind of protein called histidine kinase that sits in the membrane of the bacteria. This detects different signals, either within the membrane or in the extracellular or periplasmic space of the bacteria. When it receives that signal, it will autophosphorylate and transfer a phosphate residue to a cognate response regulator protein. And so we can sort of consider this to be a kinase transferring the phosphate group to its substrate. But the really interesting thing and useful thing for us was that these histidine kinases also tend to have phosphatase activity. So in the absence of any input, they tend to dephosphorylate their response regulator substrate. But then when we add input, the logic flips, and then they will dephosphorylate instead. Or sorry, when they add input, then it will phosphorylate. And nicely, there's actually dozens of these just in E. coli alone, one commonly used lab bacteria, and they're all, for the most part, orthogonally acting to each other. So the ones that we chose to work with are two really well-studied proteins, OMZ and OMPAR from E. coli, and we engineered them to make a kinase and phosphatase version of OMZ. So instead of having this bifunctionality, it would just have either a kinase activity or the phosphatase activity. Okay. So we plugged these in and started testing them out. The first thing that we did was just to take the kinase and phosphatase and see if, when we dose them into the cells at different copy numbers, which would give us different amounts of each protein, do we get the sort of competitive phosphorylation and dephosphorylation of our OMPAR transcription factor that we would expect to see? And does that in turn affect the expression of the output? And so what we see is that the answer is yes. So when we add more and more kinase to the system, we can basically induce the activation of the output. And when we add more and more phosphatase, we can basically suppress the expression of the output due to the dephosphorylation of the transcription factor OMPAR. And we can also tune the phosphatase stability through addition of a small molecule called TMP and infusion of some inducible destabilization domains called DDD. So in this case, when you add more TMP, it will stabilize the protein, which will lead to faster dephosphorylation. And ideally in our model, with the way that the phosphatase will be coupled into the system, this should lead to a smaller epsilon. So just to confirm that this works, we set up this system with a set amount of the phosphatase and kinase, and then we dosed in TMP, and what you can see is that it causes the system to turn off and works across different amounts of kinase, being the different colors here. So our final sort of feedback control circuit looks like this, where we have the kinase being treated as the input. This phosphorylates the transcription factor, which then induces expression of a reporter, which we consider the output, but also the phosphatase, which circles back around and completes the feedback loop. The phosphatase stability can be tuned by the small molecule TMP to tune the feedback gain. The way that these are expressed is that they're two-way linked together, which means that they're expressed on the same RNA transcript, but they're generated as separate proteins, which is not something typically done in bacterial systems, but it's something that we can do in mammalian cells to really couple the production of these two species, even if they're not fused directly to each other. Okay. So we took the system and exposed it to resource competition using one of the most potent resource competitors that we had worked with previously, called Galfor Viper. And then we looked at the fold change in the output in response to this perturbation across the whole input-output spectrum. And what we show here in green and blue are two different versions of the feedback controller. The blue one doesn't have the degradation domain here, and the green one does. And we're just looking at one particular level of TMP here, actually a relatively low amount of TMP, because if you add a lot of TMP, the system becomes very hard to turn on, actually, because the phosphatase is so strong. So anyway, we're comparing those to different open loop systems, and basically you see that initially at low levels of kinase, they respond similarly to the perturbation as the closed loop. But as we add more and more kinase, we build up more and more of a pool of phosphotranscription factor here. And so while the repression of the open loop sort of is seen, the closed loop systems, now that they have the built up level of phosphoproteins, they can actually actuate the negative feedback, and so are able to stabilize the system against this perturbation. So looking at the dose response curves of the kinase to the output with and without perturbation, since those fold changes are a little bit abstract, this is what they look like. So when you induce with the open loop system, there's a very large difference between the induced level and the perturbed level, whereas with the closed loop system, it tracks a little bit more closely, but it's still not perfect. Another nice side benefit of this is that when we induce the output here, the negative feedback does another sort of expected thing, which is to decrease the noise in the expression level of the output. And so we get a nice, more uniform graded activation of the output instead of this sort of more bimodal and noisy activation that you would observe in open loop systems. So why is it not perfect? The reason is more or less because of some incompatibility between this term here and this term here, given or for the parameters of the system that we were working with. So going back into the model, we required that the phosphatase is saturated, meaning that there's enough phosphorylated transcription factor to saturate the phosphatase so that it's operating in sort of the saturated regime. But conversely, you need that this promoter is not saturated by that same transcription factor. And so this needs to be operating in a more linear regime. So in order for that to be true, for a given level of phosphorylated transcription factor, you need that this KD is much, much larger than the K2 or the Michaelis constant of the phosphatase, which in our system, they were probably more similar to each other. Limiting the sort of dynamic range of the feedback control and thus the ability of it to adapt to perturbations. So future versions of this design will definitely have to try to fix this and maybe some other aspects of the system that might not be ideal. But for now, we were fairly happy with what we were able to achieve there. So for the last part of this talk, I'll go into our feed-forward controller, which works a bit better, specifically for the problem of resource competition, and I'll explain why. So the way that feed-forward control works again is in this case, we sort of detect directly the disturbance or the resource competition event, and then use that to offset any effect that it has on a controlled system of interest. So take a typical gene that is expressed in the cell. This is coupled to resources through its use and effect on those resources in the cell. And so any other competing modules that are introduced will load those resources and affect the circuit. But what we can do is build a sort of resource decoupled module where we have some sort of sensory gene, in this case, an endoRNase, which is a protein that will bind and destroy the target mRNA of the output, that also responds to changes in resource fluctuations. And given that the sort of problem in mammalian cells is a sort of transcriptional problem, what we do is give this gene and its gene that it's regulating the same promoter. so that ideally they're reacting to the same shared transcriptional resources that are perturbed by other genes. And so there should be a sort of ideal or maximal coupling between the regulation of this gene and this gene by perturbations to the availability of resources. So we can sort of make a shorthand cartoon of this where there's some load on shared resources that feed in both to a endoRNase. So Cassie is the one that we'll use here, and I'll explain in a second what that is, and its output. And this forms a sort of incoherent feed-forward loop, which is a fun motif that has been seen a lot in cells that allows for some outputs to be made insensitive to changes in some upstream module, at least in the steady state. There might be a transient effect, but because of this action here, there can be an offsetting effect. And so if this works well, what we'd expect is that the more that we load the resources in this system, the more that an uncontrolled system, one without the endoRNase, would go down in its expression. But with the endoRNase, we can ideally keep the expression closer to the target. To implement this, we used an endoRNase derived from CRISPR systems. So Bree was a graduate student in Ron Weiss's lab at the same time as me, currently at A2 Therapeutics. And she went through and screened and found a nice library of endoRNases from CRISPR systems that work well with regards to, or that work well in mammalian cells, basically. So the assay that she did was basically just to express different endoRNases in cells with different reporters that had different target hairpins for different endoRNases. And so if the endoRNase can recognize and cleave this target hairpin in the RNA of this reporter, then it will prevent protein expression. And so what you're seeing here is the fold repression of different reporters by different endoRNases targeting different hairpins. And for the most part, these work nicely orthogonally to each other, meaning that they target their hairpin and they target very few other hairpins from other systems. So the one that we chose to work with was one called Cas-E, which had overall fairly nice behavior and was one of the highest fold repressions. We avoided using this one CSY4, which is maybe slightly more active, just because it seems to have more toxicity or off-target effects in the cell, whereas Cas-E is a little bit more well-behaved. So the implementation of the system looks like this, where we have the endoRNase being produced. So Cas-E, it binds to this hairpin in the output reporter. They both are being driven by a version of the CMV promoter. And notably, we can tune the system, tune the amount of endoRNase that's being made by adding upstream open reading frames, which are basically short sequences that will divert the ribosome away from this coding sequence. And so the more of these upstream ORFs that you have, the less expression of the endoRNase that you will have. So in our model of this, the more free resources that you have, the sort of more output that you should get. Sorry, back that up. So the system is basically able to to rescue this the level of output through some threshold level of resources. So if the level of resources is very high, then your output level is fine. But if you decrease those, the system, when it's sort of working well, can actually maintain the level of output until you reach some critical threshold, where then it will no longer work. And so this threshold is proportional to, again, some value epsilon. And that epsilon has to do with both the catalytic rate and the expression level of this enzyme. So the more of the enzyme that there is, and the faster its catalytic rate, the smaller the epsilon. So you'll shift this threshold to the left, and the system will become more robust to more and more depletion of cellular resources in terms of the expression level of the output. But we will note that if you decrease epsilon, you also decrease the output level. And so there's a little bit of a trade-off there. Okay, so I think I got slightly ahead of myself, but the epsilon here is basically inversely proportional to the catalytic rate and proportional to the Michaelis content of the enzyme, and inversely proportional to the concentration of the enzyme, which can be tuned in our system by the number of UORFs. So each one of these lines here is a different version of the unregulated system without the controller, where we just tuned the copy number of the output so that we could make sure that just the set level of expression wasn't affecting the ability to be repressed. And what you can basically see is that as we add in more and more of the resource competitor, Gal4 Viper, that we deplete the resources and decrease the expression of the output, regardless of what the initial expression level of that gene was. When we have the controller, it works quite nicely. So it's able to prevent resource competition from affecting the level of output. But then as we add more and more UORFs, we basically decrease the expression of Cas-E, which increases this value epsilon and sensitizes the system to the repression of resources by sequestration of resources by Gal4 Viper. So with this experiment, we're able to sort of validate our sort of model here of what we expected was going on with respect to the parameters of the system. And so if we take a look at this one here, and then just one of these that starts at a similar expression level in the absence of perturbation, so that would be the first boxes here. So we add in Gal4 Viper, you can see here more precisely the decrease in the level of output, and that is observed with the open loop system, but not seen when we have the controller. Okay, so an interesting thing that we noticed with this is that if we put this controller into different cell lines, it actually really precisely regulates the expression of the output across these different cell lines. So over on the left is an experiment where is actually from that experiment where we tested out different promoters and different cell types. And in that case, each of these different promoters doesn't have the same level of expression in a different cell. So basically you have a lot of variability, and it's hard to predict based on the expression in one cell type, what you're going to get in another cell type. Whereas with this control system, we can actually get quite similar levels of expression across different cell types. And the different levels here are being set by the different numbers of UORFs in front of the endoRNAs. And so it seems to be able to insulate the environment of the cells or insulate the gene expression to different sort of intracellular environments that it's placed in, which was, well, it's kind of what we would hope to see, but it was nice to see it in action. And then when we tested out the ability to react to different transactivators placed in these cells, we can see that with the controller, we're able to basically prevent a lot of the repression of output that we would see when we don't have the controller present in the cells. And these different varieties here are just different unregulated and regulated systems with different initial expression levels. And so we're able to maintain expression levels and robustness of those expression levels across different cell types. So in summary, what I've talked about today is the pervasiveness and challenge of dealing with lack of modularity in systems, in particular cell resources, which can have different effects and strange effects on the expression of your system, whether it's in bacteria or mammalian cells, and how control devices, both feedback and feed-forward controllers, can be used to detect and adapt to perturbations such as resource sharing and competition. Our sort of vision moving forward for using these systems is that we can take, for example, and this is what I'm working on now in Peter Zanstra's group for my postdoc, is can we take, for example, pluripotent stem cells, and can we control their development towards some target type of cell. So for example, a T cell that would be used in therapy. So typically this is done through just controlling the media or other factors that the cells are sort of experiencing as they differentiate. But in the future, perhaps we can actually detect the state of the cell as it's differentiating and then use control systems to guide their development more precisely. Similarly, once we have developed these therapeutic cells, perhaps we can use feedback control systems to regulate their function when they're used in for treating a patient or for targeting a cancer or something like that. Right now, we just put cells into the body and we kind of hope that they work. But there's so much more sophisticated control that we could do to really ensure that cells are reacting to their environment, for example, the tumor environment in an appropriate way. And this would be important both for patient health and safety and also for the success of the therapy. So with that, I'll just say thanks to everyone that helps work on these various projects, highlighting here some of the people that I mentioned along the way. And to our funders for these projects, my current funding, to the departments that I was a part of while I was at MIT, and again to everyone here for listening and to the organizers for dealing with my time zone disruption. So with that, I'll be happy to take any questions.
Thanks a lot, Ross, for this great talk. I start perhaps with the room here. Are there questions in this room? Mike, what? Otherwise, in the meantime, feel free to raise your hand and unmute yourself in the Zoom, or write a question in the chat if you cannot unmute yourself, and we will read it for you. Of course, yeah. I see one raised hand from Manish. So Manish, please.
Yeah, hi Ross. This is a great talk. Thank you for walking us through all these different designs, which to me seem very, very challenging, and there's lots of work here. I was wondering, for your sort of mammalian control circuits, are these all genome integrated devices? So they live at single copy number, and do they all go into the same place in the genome, or do they go in different places? And also, what is I'm guessing, I mean, the data you present is sort of steady state-ish data, but what is the response time for these circuits to accommodate to perturbation, given part of the circuit is operating in the cytoplasm and the genome is of course in the nucleus, and there is shuttling of molecules between the two. So do you have any thoughts on that?
Yeah, that's a great question. So we didn't integrate these into the genome yet. We're definitely interested in doing that. At the time, we were just doing transient transfections and going through as many of these designs as we could to get something that would work right. So they're uncoated on plasmids. The plasmids are transiently delivered, but within that transient delivery period, the system can achieve somewhat of a steady state. It's certainly possible that if it was integrated, that there would be more time for the system to achieve steady state, which might actually be beneficial for I think for the feedback controller in particular, because there's not a lot of time for that to react in the time window that we give it. We did try to look at this a little bit in this sort of transient context, the response time, but we couldn't quite figure it out just because of the complexity of the transfection process. But we're certainly interested to see, yeah, if it was integrated and then we put it in that perturbation and then imaged it over time or something, how fast would it react? I think that it probably wouldn't be like a super fast response time of the transcription, because you're still feeding back through a transcriptional process. And so you do have to wait for the transcriptional delay and all of that to sort of work its way through the system. The key thing for us, like mathematically, is that the controller species, the phosphatase, has a very fast reaction in terms of its actuation on that transcription input. But certainly it would be probably preferable to have a system that was maybe wholly working at the protein level. And actually, this is a very similar design to what sort of naturally exists in a lot of signaling pathways, where you have a phosphorylation cascade, and then at some point there will be either a kinase or a phosphatase that will go back and then modulate upstream pathway activity. And so those systems will act very fast, and they'll also have this type of negative feedback circuit built in. So we're definitely inspired by that, and maybe in the future we can make some like completely protein level circuits, but I think that there will be a considerable delay in sort of the reaction time to any perturbation just due to the transcriptional layer.
And with these transcriptional circuits, do you anticipate that when they're in the genome, there will be effects of chromatin remodeling that will kick in as well? And might that complicate the response time in some sense?
Yeah, definitely. We would have to put it into some locus, like safe harbor loci, where the genome is open, because we would need it to react to the transcription factor. It doesn't have to be a linear reaction to that transcription factor. It just needs to be able to respond to that transcription factor. And if you have a locus that started to close up, then the transcriptional response, it might not work, or it might be delayed or something like that. And so, yeah, that's definitely one of the considerations we've been thinking about. And one of the reasons we avoided going into the genome right away is because we didn't want to have to deal with that kind of thing while also developing the circuit. But when I think about, for example, putting this into a developmental context or into a cell that would be used for therapy, then it absolutely has to be in the genome, and you're going to have to fight against this silencing problem in addition to any sort of other chromatin effects. And so I think it's like somewhat of a different problem, like trying to deal with that. But yeah, we need to work within the confines of that chromatin context for sure.
And may I so on the point of the transient transfection, then, does that mean that you have different copy numbers of DNA delivered into cells, and therefore different cells will have effectively different circuits because of the dosage being different?
Yeah, exactly. We actually have a reporter, or so transfection reporters you can use to sort of determine what is the delivery. And so you can isolate cells that have set amounts of that reporter, and then we actually evaluated how the circuit was performing across all kinds of different combinatorial dosages of the reporter and the phosphatase and other molecules in the system. And so we like systematically went through that to see what it would do. And what I show is just some subset of those results. But there's definitely regimes where based on how much relative expression of the phosphatase and the reporter and so on that you have, well, those are technically on the same plasmid, but like the relative amount of the kinase and the relative amount of the reporter / phosphatase and so on, there are optima there where the system works ideally. You can't have like too much kinase, otherwise, well, we use the kinase dose response as it is, but at like no kinase, the system doesn't work because there's no dephosphorylation that's possible. And then at sufficiently high kinase, there's it's like already saturated basically.
Okay, thank you.
So we have perhaps time for one question. Xio Yan, do you want to unmute yourself? Or was okay, perfect. Good.
Hello, can you hear me?
Yeah, it works.
Hi, Ross. That's a really fantastic talk. I really like it. I have actually two questions regarding the feed-forward controller and negative feedback controller. You briefly mentioned that the feed-forward controller is better than a negative feedback controller, right? So do you want to explain why, or what is the mechanism why the
yeah, just in this case, I won't claim it always, but it seems to work well here. I think basically it's a problem of parameters. So in the feedback controller, we have a slight issue getting the parameters right to where we can saturate the phosphatase and not saturate the reporter. And we did try to play around with that a little bit. I didn't go into detail by, for example, using mutated versions of the promoter and the activator, but it was hard to get that tuned right. And so we just went with what we had there. With the endoRNase, with the feed forward control, it's pretty easy to get the math right, so to speak. So you need high activity, high expression feed forward molecule, in this case, the endoRNase. and they have a really fast catalytic rate. They're reasonably stable in the cells, so you can produce a lot of the molecules, and yeah, because of that, you can get a good effect. It's also important, I didn't mention, that the effect is linear. So some feed-forward circuits that people have tried to make using transcription factors have been sort of hampered by this in the past, because the transcription factor might have some cooperative behavior where it binds and regulates the target gene. But with endoRNases and microRNAs, which is another sort of RNA level implementation that Mustafa's lab did, you can get basically that nice linear effect. So if you have an increase of one endoRNase molecule, then you'll have a decrease in one sort of target RNA molecule. It's proportional in that way. So for us, that means that we get a really nice proportionality between the response of the endoRNase to resources. So if the endoRNase goes down because the resources go down, then it will go down by the same amount that the output will go down. And then because of the proportionality, that means that you'll have an offsetting effect. I will mention that there is still this problem of growth feedback. The resource competitors can decrease the growth rate of the cell, both in bacteria and in mammalian cells. And our endoRNases don't necessarily have the same decay rate or degradation rate as the target gene. And so actually there were some instances where we saw the output kind of go up in response to resource competition in a way that was consistent with what we'd expect, given that the endoRNases are probably less stable than the fluorescent reporters that they were regulating. So there are definitely some things that can still go wrong, but as far as the system here that we're doing with resource competition, it seems to get the job done in a satisfactory way, I guess I'll say.
Okay, thank you. Another question is about the difference between the mammalian cell and the bacterial cells. You mentioned that for the bacterial way, the resource competition is more for the ribosome, right? But for the mammalian cell, it's more for the co-activators. So based on the difference, do you think I don't know, when we design the controller, should we consider this difference and design a different controller, or what is your perspective for this?
Yeah, that's a great question. So it matters definitely for the feed-forward control. For the feedback control, you could feasibly design a similar circuit that would work for both. So the benefit of feedback control is that it doesn't really matter what the perturbation is. You're just detecting the output, whatever it is, and then you're fixing it. And so that's why we still are very interested in getting this to work really nicely. Like, for example, the feedback control systems that Mustafa has been working on lately. If you can get the parameters right, you can get it to adapt to any perturbation that produces that output. But in the feed-forward control case, our design wouldn't necessarily work for ribosomes. It might work for ribosomes. I have to think through this for a second. So technically, the endoRNase production is dependent on ribosomes. The output is dependent on ribosomes. So I think if the ribosomes are depleted, it should work for them as well. But it's a little bit trickier because the output is sort of being expressed downstream of the ribosome, or downstream of the action of the endoRNase, right? So the endoRNase regulating the output will also affect the ribosome usage. And so it gets to be a little bit more tricky. I think that you might need for feed-forward to work with ribosomes to do like protein level regulation instead. Because right now there's sort of an implied feedback loop where the ribosome would affect the endoRNase and the output, and then that would affect the ribosome further once the endoRNase reacts and changes the translation rate of the output. So yeah, I would go for some protein level feed-forward loop, like using, for example, maybe those like programmed proteases or something like that instead.
Sorry to move on because we're a bit running late in time. But Ross, if you're around in Zoom for a few minutes, people can just feel free to write in Zoom chat. Yeah?
Oh, yes, I'm happy to respond on the chat.
Yeah, thank you.
Everyone for the great questions.